All right, in this video, the goal is to talk about a concept that is very similar to the last video, which was discussing using antibodies as a form of medication. So, as a reminder, in the previous video, we talked about using antibodies in the sense that the antibody is the medicine, right? So you take an antigen, usually something on a cancer cell, or maybe a protein and like a plaque in an Alzheimer's brain, and you put that antigen into a mouse, and you get the mouse to make the antibody for you, and then after some modification, put the antibody in the patient. Hopefully it binds to a target and either blocks the activity of that target or stimulates the immune system. The idea of a cancer vaccine is very similar to that, and the difference is that instead of making the antibody right in an animal ahead of time, we're actually going to find a way to get the patient to make the antibody themselves. And so the way this works is, in a typical cell, we assume the cell is normal and healthy. Remember, DNA encodes RNA, and that RNA forms some sort of protein that's made up of amino acids, right? And that finished protein, we hope, looks completely normal, right? That means that if the finished protein were to find its way to the surface of the cell, this T cell would just ignore it because normal, healthy protein, no mutations, it's recognized as being a self antigen, meaning normal part of the person, nothing to worry about. In a mutated cell, even very, very small changes can potentially be detected by the immune system. And so this concept of a neoantigen, all that means is if there was a mutation in your DNA, it's going to get transcribed into the RNA, and that's going to get translated into the protein. So even if you only have a single amino acid difference, that can be enough that a novel either antibody or this would be a T cell receptor, either way, those molecules in the immune system are now able to recognize that modified antigen because it's just different enough from the normal one that it's going to be marked as foreign, right? And if this is one of those CD8 positive, right, cytotoxic killer T cells, the end result is going to be T cell is going to eliminate the cancer cell with the mutation because that's their entire job. These T cells are on the hunt for either cells that have viral infections, in which case you'll get viral proteins on the surface, or cancer cells that have mutated human proteins, again, that are just different enough that they look foreign and we can tell them apart from the healthy version of the cell, right, like it normally would be. All right. Now, the way that you end up doing this, we've got a few different options. The thing that you've got in common is you have to figure out what your target is, right? So in this neoantigen discovery, that simply means we're searching in what can feel like a needle in a haystack. There are tens of thousands, hundreds of thousands, really, of different proteins inside of a human. And not all of those proteins are expressed in every cell. And so our first question is, all right, what proteins are expressed in the cell? This idea of a transcriptome means all the genes that are expressed in that particular cell type, right? Not necessarily all the genes in the entire human genome. We're only concerned about the ones that are used in our cancer cell. And from there, what you're looking for is any alteration from normal. Okay, so when we say that we're looking for a neoantigen, you're going to take that transcriptome from the cancer cell and compare the sequences that you get back, right, the DNA and RNA sequences, to a normal healthy cell that's of the same cell type. So for example, if we were looking for a cancer vaccine to treat liver cancer, we would look at all the genes expressed in normal healthy liver cells. And then separately, we would look at all the genes expressed in this liver tumor, right, from our sample, and start hunting for differences. The differences are the neoantigens. All right, at that point, you know what your antigen is going to look like. The question is, how are you actually going to deliver it into the patient? And you've got a handful of options. The first is you can actually inject mRNA or DNA directly into the person. This is the same concept as how the COVID vaccine works. You're not giving COVID proteins to the person, right? You're giving COVID mRNA, and then the human cells are copying that into proteins that stimulate the immune system. All right, option 2, these SLPs stands for synthetic long peptides. That basically means making the protein directly. So taking the mRNA and DNA, converting it into a protein peptide sequence, injecting that directly into the patient, that is the more direct route. The mRNA and DNA has the advantage of being slightly longer lasting because you're giving the person the ability to actually make the protein themselves. All right. Pulsed DCs, these are dendritic cells. This is where you actually take cells, these dendritic cells are part of the immune system in the patient, expose the dendritic cells to these peptides, and then put the actual whole cells back into the person. That's effectively getting even closer to the final product, right? Because what we hope happens is if you're using either of these routes, you inject those into the patient makes a protein. Eventually, that's going to find its way into lymph nodes where B cells and T cells live, and that is where activation is going to happen, right? These T cells are going to see the antigen on, this is our antigen presenting cell. So you can give the person the DNA or RNA to make the antigen. You can give them the protein that actually is the antigen, or you can take dendritic cells out of their body, sort of prime them by exposing them to the antigen ahead of time, and then putting those activated dendritic cells back in the body as well. Okay. And then lastly, these nanovaccines, that's really just more of a technology thing. Nanovaccines means whatever molecules we're going to give, whether it's protein or DNA or RNA, can be packaged into these very tiny, like submicroscopic nano vesicles, these little lipid-bound vesicles. They just increase delivery to make it easier to get those DNA, RNA, or protein molecules into the patient and into the target cells that we want them to affect. All right. So this summarizes a little bit more clearly just the delivery strategy, right? We can give nucleic acids that code for the protein. We can give the protein itself, right? We can use a virus. That's an alternative delivery system where you're still delivering DNA or RNA. You're just using a relatively harmless virus as a way to actually get it into the patient. Or lastly, cell-based vaccines. This is the dendritic cell thing that we saw before. So these are all options. They all result in the immune system attacking a certain antigen. Okay, and in all of this, just remember, the delivery is relatively straightforward. Once we know what antigen we're going after, that can be done. Figuring out what the antigen you're going to use actually is, this is the part that is a huge question mark because there's no guarantee that you're going to find a good one. Different cancer types have different mutations, and you're sort of hoping that you're going to find the best possible option, but it's hard to say exactly how good that antigen will actually turn out to be as far as how good it is at activating immune cells. All right, so as an example of this, like we said, there are different molecules you can use, there are different delivery mechanisms. This is a good example of how this system can be customized very easily. So in this case, we're using an mRNA-based vaccine. Right, again, same concept as the COVID vaccine, only now we're not targeting like a viral antigen from COVID, we're targeting cancer antigen. And LNP is this lipid nanoparticle. All right, the lipid nanoparticle, the shell of it is made out of lipids that look very similar to what human cell membrane lipids would look like. And then we pack it with our RNA molecules, right? This is our RNA on the interior, right? That RNA was very, very specifically designed to code for the neoantigen that we found during our screen, right? And the hope is that once we inject this into our patient, that nanoparticle should be recognized by the cell as looking like something human, taken in, and that mRNA is going to be released into the cytoplasm of the cell, where it will go through translation just like any mRNA would. And the end result is our antigenic protein. Remember, this is that neoantigen, right, that we selected out of our screen because it is unique to our cancer. Right? Now, that antigenic protein can then be exported up here and displayed on the cell surface so that T cells that are out looking for anything suspicious that might be foreign will recognize it, and that tips them off that this cell is either cancerous or has a viral infection or whatever it might be. And so we're basically just taunting the cell into making the antigen that we want to prime the immune system, right? And now those T cells will see this and go out to the rest of the body hunting for it wherever they might find it, right, on our tumor. Okay, one of the problems, I guess I shouldn't say problems, one of the more difficult aspects of this is remembering that these types of therapies are not immune, as it were, to the problems that cancer therapy has in general. And probably one of the most difficult aspects of cancer therapy is trying to find treatments that address as many different possible variations of the cancer cell as possible. What I mean by that is, in this picture, they're trying to show us that this tumor is heterogeneous, right? The different colored cells in here, their key is showing us that these cancer cells have mutations that are sensitive to whatever therapy we happen to be working with. This is all very hypothetical. And so if you give the treatment, notice you've eliminated most of the cancer cells that fit into that type, right? But these other types, right, that have a different set of mutations, they're going to be resistant and they're going to thrive. And so this explains why with a lot of cancer therapies, whether it's small molecule like traditional chemotherapy or cancer vaccines or antibody-based therapies, whatever your drug is targeting a particular mutation or protein inside that cancer, right? And the reality is you are very, very unlikely to find a treatment that is going to kill every single cell in the tumor. It's probably not going to happen, right? So if you can find treatment number one where the cells are sensitive to, right, that drug, maybe you have drug number two and the tumor cells are sensitive to that drug, and then hopefully maybe drug three where they're sensitive to that one. In sequence, you can string all these drugs together. One of the hopes of this mRNA-based therapy is that you're not limited necessarily to a single RNA, right? So if we use this lipid nanoparticle, there's nothing that says that you have to use only a single RNA for a single antigen, right? The concept of making combination vaccines hasn't really been put into practice clinically yet, but the hope is that we can maybe combine 2 or 3 or 4 different RNAs for multiple antigens. That way, you're effectively delivering multiple drugs all at once, priming the immune system to make, antibodies against every cell in the tumor, not just a small subset. Okay? Because otherwise, eventually, you're going to get a resistant tumor that is not going to respond to any of these drugs. And the more rounds you go through, the more and more difficult it is to find therapies that are effective. Okay, so as an example of this, this paper shows a few different things that I think are very interesting. The first is we are using an mRNA-based cancer vaccine, just like we've been talking about. The second interesting part of this is that it is personalized in the sense that, remember, no two cancers, or very, very rarely, are two cancers identical. And so if you find 2 patients that have the same type of cancer, right, liver cancer, breast cancer, even more specific types, they're going to have different mutations, right? They might even have different mutations in the same protein, right? So thinking back to the beginning of all this, right, you might have a series of patients that all have a mutation in a certain gene, okay? But different patients might have mutations at different points of that gene. And so it might be different amino acids, right, that have been modified. If that's the case, then the neoantigen from one patient to the next is going to be slightly different, right? So you can't take the exact same RNA and use it across the board in many different patients because their cancer has a slightly different alteration to the protein that we're trying to target, right? Now, if you can make it personalized, meaning you collect a tumor sample from every individual patient, you sequence the DNA or RNA in those cancer cells and find out exactly where the mutations are. You effectively custom make an RNA specifically for their particular kind of cancer and the mutations that they have. All right, so the general concept stays the same. The delivery mechanism will stay the same, right? Those lipid nanoparticles. But the exact specific sequence of the RNA is adjustable and personalized. So the upside of this is it probably has a greater likelihood of success in the long run because of the personalized nature of it. It also probably has the prospect of being much more expensive because you can't mass produce in the same way that you can with a small molecule drug, given that every patient is going to require a slightly different, personalized sequence. Okay, so as an introduction to this, so the type of cancer that we're using as an example in this particular set of data, this resected melanoma. Melanoma is by far the most aggressive form of skin cancer, and it's known for being very, very difficult to remove. Right? And so this data this is considered this background. So alive and recurrence-free basically means percentage of people starting at time 0 after their first round of treatment. And that means they've survived round 1, the tumor's been eliminated, but these melanoma cancers are known to sort of come roaring back and cause problems again. And so over the course of up to 6 years, we're measuring the ability of, right now, this is a monoclonal antibody, pembrolizumab. That is an immune checkpoint inhibitor, right? Very similar to the immune checkpoint inhibiting antibodies that we talked about in a previous video. They are significantly better than a placebo, right? So for example, at this 5-year mark, which is a significant amount of time for this type of cancer, we go from somewhere in the upper 30s, low 40s survival rate up into the 50s, approaching 60, right? So not magic by any means, but a pretty significant increase. Right? What we're wanting to know now is, can we combine that customized mRNA vaccine in addition to this immune checkpoint inhibitor as a way to, right, even further improve outcomes? Okay, so this is showing their process. Remember, this is individualized. So they have to go through this process separately for each individual patient, right? We're taking a tissue sample, tumor versus normal, to make a comparison, right? So they would take melanocytes from normal, healthy skin, and then melanoma cells from the tumor, sequence them to identify mutations that have the potential to give neoantigens, take those hits, turn that neoantigen into an RNA that can be administered to the person, hoping that they produce the protein which stimulates the immune system to go throughout the body and find the tumor, right, wherever that antigen is present. Okay, so we sequence data from the tumor, or DNA or RNA. We feed that data into this algorithm that selects our best neoantigens available. Right, we use that to make an RNA sequence, and in the end, they found 34 different neoantigens present in their patient sample that they made, right, to use as an adjuvant or an additional therapy on top of the immune checkpoint antibody drug that's considered standard of care. Okay? So in the end, we expect it to look like this. Those mRNAs that were made, again, customized to each individual patient, are loaded into our lipid nanoparticles. That's these guys, injected into the patient. We hope those fuse and enter cells, get translated into a protein. Those proteins are going to be recognized as at least slightly foreign, right? And our neoantigens that are not recognized as normal human proteins are going to get exported onto the cell surface and shown to the immune system, where they're going to act like a stimulant, right? That's our hope. And in order to test this and see if it really works, right, to find out whether these T cells are going to go around the body and attack tumor cells the way that we want them to, we have to do a comparison, right? But The catch is we already know that this pembrolizumab, this is our checkpoint inhibitor. That was our standard of care from before, right? And this is amount of time, right, in weeks during this trial. So this risk-free, regression-free survival, we start at 100% at time 0, and we just give both combinations, or both drugs, the immune checkpoint inhibitor by itself, the bottom line, or immune checkpoint inhibitor plus our customized mRNA vaccine. And at their different time points, we see significant numbers, right? 62.2 versus 78.6. That is a significant difference at that 18-month time point, right? And that difference is maintained pretty well through the entire study. There's kind of a sharp drop-off at the end, but during this time frame, that is making a fairly large difference for a pretty good number of people. Now, an interesting question from all this is asking, can we do this with multiple antigens? Meaning, when we go through this screening process, it is entirely possible that you could find more than one antigen for each patient. Clinical trials are built around the idea that you only test one variable at a time, but it's not unreasonable to think that we couldn't identify as many different antigens as possible to combine them into a combination therapy. And maybe if we target more of those antigens, we'll increase the likelihood that we stimulate that T cell. And hopefully this line will continue to get higher and higher, right, as those improvements are made. All right. Hope you found the video interesting, and check out the playlist to make sure that you get right, all the videos that are added.